Your DNA is like an instruction book. It contains the instructions for living things like you to develop, grow, and functions. Variations in DNA instructions can make a difference to how a living being looks, develops, or responds to its environment. The complete set of a living being's DNA instructions is called a genome. Our human genetic instructions crammed into our cells are made up of more than 6,000 millions DNA letters that are divided across 46 chromosomes from our parents, 23 pairs from our mother, and 23 pairs from our father. The letters represent chemicals along the tightly packed strands of DNA is known as A, C, G, and T. The order or sequence of the DNA letters is important. The cells of our body use sections of DNA sequence called as gene as templates to make proteins and other molecules you need to function. Genes can be anywhere between hundreds and millions of letters long difference known as variants in our DNA sequences contribute to our individuality and affect the way our bodies functions. Variants in genes can change the gene product that is made. This can cause it not to work as it should or not at all. Variants outside genes can change how and when genes are used. You have two copies of most of your genes, one from your mother and one from your father. So you can inherit variants from either or both of your family. Some variants can affect your health and the health of your family. What is single gene disorder? You can see a chromosomal of a somatic cell. There are 22 pairs of autosome and one pair XX or XY. Now, it's focused on a single chromosome. For example, if we focus on chromosome 7, we can see that there are 900 to 1,000 genes in this particular chromosome. We know that some genes are dominant and some are recessive by character. The arrow indicates a gene known as CFTR. It is a recessive gene. Mutation in this gene is responsible for a clinical disorder known as cystic fibrosis. I think now you can understand what is meant by single gene disorder. Again, single gene disorder comprises a spectrum of certain genetic disorders. We have autosomes and 6 chromosomes. Again, we have genes of dominant and recessive characters. At first, we will understand the mechanism of inheritance. Then, we will see some examples. Please look at the picture. The arrow indicates mutation of a dominant gene. The sons and daughter who will inherit the single copy of that mutated gene will be affected. Since the copy is dominant, it will express. In this pattern of inheritance, both males and females are equally affected. Usually, 50% offspring have the probability of being affected. Again, both parents may be normal, and the offsprings can get a new mutation. So, this is another way of having autosomal dominant disorder. Now, we will see some examples. Huntington disease is a neurodegenerative disorder mostly affecting the basal ganglia. Marfan syndrome is a connective tissue disorder. Usually, the patient has large stature and long arms and fingers. So our next category, X-linked dominant inheritance. In this picture, you can see father has got mutation in the dominant gene of X chromosome. Now, what is probability of being affected among his offspring? Here, we need to consider two things. Number one, dominant gene will always express. Another is Sons do not receive X chromosomes from father. Only daughters receives X chromosomes from father. So, sons will not be affected and all the daughters will be affected. So, let's consider another scenario. Okay, here, mother has got the mutation. Now, what will happen? So, sons and daughters both receive X chromosomes from mother. So, sons and daughters who will inherit the mutated gene will be affected. One example of the start of disorder is Fragile X syndrome. The child usually has developmental delay and learning disability.
Chromosomal disorder. Chromosomal disorder is any syndrome characterized by malformation or malfunction in any of the body system and caused by abnormal chromosome number of constitution. Might be an excess or deficiency of the whole chromosome, chromosomal rearranged and contiguous gene syndrome. Because the chromosome abnormalities can be numerical or structural. A numerical abnormality means an individual is either missing one of the chromosome from a pair or has more than two chromosomes instead of a pair. A structural abnormality means the chromosome structure has been altered in one of several ways. Always remember that the disorder is a random error in cell division that happens when a parent's reproductive cells are being formed. A set of chromosomes, as seen under a microscope, is known as a karyotype. So, what is karyotype? Karyotype is a test to discover and evaluate the size, shape, and wide variety of chromosome in a sample of body cell. Extra or missing chromosome or abnormal position of chromosome pieces can cause problems with a person's growth, development, and body function. Any deviation from the normal karyotype is known as a chromosome abnormality. Chromosome abnormalities commonly occur when there is an error in cell division, resulting in cells with too few or too many copies of a chromosome. An error in cell division called non-disjunction results in reproductive cells with an abnormal number of chromosomes. Klinefelter syndrome. The abnormalities is XXY. This Klinefelter syndrome is born with an extra X chromosome. Instead of the typical XY chromosomes in men, they have XXY. So this condition is sometimes called XXY syndrome. This kind of syndrome only affects males. Not all boys with Klinefelter syndrome will have noticeable symptoms. Other boys can have symptoms that are physical, apparent, or problems with speech, learning, and development. Babies with Klinefelter syndrome typically have weak muscle, reduced strength, and quiet personality. They also can take longer to do things like sit up, walk, and speak. So, the signs of a Klinefelter syndrome are taller, less muscular body than male their age, broader hips and longer legs, large breasts, weaker bones, a lower energy level, smaller penis and testicle, delay in puberty or go a parcel amount, and less facial and body hair following puberty. There is no cure of this syndrome, but it can be treated. So, how is Klinefelter syndrome treated? There's no way to change the XXY condition if a boy is born with it. But treatments can help relieve some symptoms. As with many conditions, beginning treatment early can make it much more effective. Turner syndrome. Abnormalities is monosomy X. Gonadal dysgenesis is a condition characterized by short stature, and ovarian dysgenesis patients are females with either a missing X chromosome or an abnormality of one of the X chromosome. It occurs in 1 in 2,000 to 1 in 5,000 female live births. The sexual immaturity is girls with an absent or structurally abnormal second sex chromosome. Amenorrhea. Amenorrhea occurs in patients with Turner syndrome because of absence or limited ovarian function due to inappropriate development or premature ovarian failure. Infertility. Most women with Turner syndrome have ovarian dysgenesis. Therefore, they are usually infertilized is caused by the failure of the ovaries to grow properly. Short stature. Statures mean a person's height. Almost all females with Turner syndrome grow more slowly than their peers during childhood and adolescence, resulting in an average adult height of 4 feet 8 inches. Somatic abnormalities. Many infants with Turner syndrome are born with heart defects or even slight abnormalities in heart structure that increase the risk of serious complications. So, this is the result of having a Turner syndrome. Turner syndrome can cause a variety of medical developmental problems, including short height, failure of the ovaries to develop, and heart defects. Heart defects often include problems with aorta, the large blood vessel that branches off the heart and delivers oxygen, bring blood to the body. Down syndrome abnormalities trisomy 21. The man or woman has 3 copies of chromosome 21 instead of the normal 2 copies in all cells. This is caused by abnormal cell division during the development of the sperm cell or the egg cell. This syndrome occurs because of the extra copy of chromosomes 21, which can cause the body and brain to develop different than a child without the syndrome. One factor that increases the risk for having a baby with Down syndrome is the mother age. Women who are 35 years old when they become pregnant are more likely to have a pregnancy affected by Down syndrome than women who become pregnant at a younger age. However, the majority of babies with Down syndrome are born to mothers less than 35 years old because there are many more births among younger women. That's why this kind of syndrome is one of the most common genetic birth defects. And this is the identification of the baby having Down syndrome. A child with Down syndrome may also need extra help or attention in school, although many children are included in regular class. Patau syndrome abnormalities trisomy 13. Trisomy 13 is a genetic disorder that your baby gets when they have an extra 13 chromosome. In other words, they have 3 copies of their chromosome 13 when they should have just 2. It happens when cells divide abnormally during the reproduction and create extra genetic material on chromosome 13. This is the result of the babies suffering from Patau syndrome. Babies with Patau syndrome grow slowly in the womb and have a low birth weight, along with a number of other serious medical problems. This kind of syndrome has no cure. The treatment focuses only on the baby's symptom. This can include surgery and therapy, although depending on the severity of your baby's issue. Some doctor may choose to wait and consider any measure based on the chances of your baby's survival. Babies born with trisomy 13 rarely live into their teens. Trisomy 18, also called Edwards syndrome, is a chromosomal condition associated with abnormalities in many parts of the body, caused by the presence of all or part of an extra 18 chromosome. The majority of people with the syndrome die during the fetal stage. Infants who survive experience serious defects and commonly live for a short period of time. According to Terry Kreutzer, there is a oldest woman in USA with trisomy 18. She is Megan Hayes. She still celebrates her 48th birthday. Megan survived because she was born with a healthy heart. Some doctors have realized that with the right medical care, people with trisomy 18 can live longer than previously thought possible.
Multifactorial inheritance, also called complex or polygenic inheritance, caused by a combination of environmental factors and mutation in multiple genes. The term polygenic can have a different meaning, including genetic effects that arise from the interaction of multiple genes. It can be said that polygenic inheritance involves conflict traits that are determined by many genes at different loci without the influence of the environment. Effects of those genes are cumulative, in that no single gene is considered to be dominant or recessive to one another. And here's the example of complex traits: high blood pressure, arthritis, diabetes, cancer, obesity, and heart disease.
Gene therapy. Gene therapy method of treating or preventing diseases by replacing, repairing, or returning on or off genes of a patient's cell. The process includes the use of vectors and non-viral methods to move a DNA or genes to one cell or another. And here's the video for you to able to understand what is gene therapy.
Genes are segments of DNA that direct the cell to produce proteins that perform a vast array of functions in the body. Many diseases have a genetic basis, meaning that mutated genes provide incorrect instructions that cause the cell to produce an abnormal protein, or disable production of a protein completely, either of which can cause disease. In gene therapy, a functional copy of the gene is delivered into a patient's own cells. Normal protein that is produced from a functional gene has the potential to correct the underlying cause of the disease and provide restorative therapeutic benefit. Gene therapy uses engineered viruses to deliver genes into cells. These are called viral vectors. Vectors made from adeno-associated virus, or AAV, are especially well suited for treating retinal diseases. AAV is a small, simple virus that elicits only a weak immune response and has never been shown to cause disease in humans. In gene therapy, the two native AAV genes are removed and replaced with DNA that contains a functional gene with instructions to produce a normal protein. The DNA also contains a promoter, which drives production of the functional protein. The promoter and gene are flanked by signals called inverted terminal repeats that allow the DNA to be packaged inside the protein shell of the AAV vector. AAV vectors are delivered to cells in the retina by routine ophthalmic delivery methods, such as intravitreal or subretinal injection. Once administered, AAV vectors enter the patient's cells and deliver a copy of the functional gene directly into the nucleus. Once inside the nucleus, the functional gene can support the production of the normal protein and correct the underlying cause of the disease. Genetic defects in the eye can cause vision loss or blindness, and gene therapy can provide a long-lasting therapeutic benefit.
Gene therapy holds for treating a wide range of diseases such as cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. Somatic gene therapy, those who are not related to reproductive gene therapy. The introduction of genes to cell in a target organ to produce enzymes, which are needed by the body to function well, doesn't alter the genetic makeup of an organism. And here's the video of somatic gene therapy. Reproductive gene therapy, the introduction of correlative genes to sperm cell, egg cell, or even zygotes, 4-day-old. Main goal is to eliminate the abnormalities that can possibly be transmitted to the offspring. The alteration of genetic makeup of the offspring by targeting the abnormal genes in the parent sex cell. In this, DNA is inserted into the reproductive cell, eggs or sperm, in the human body. Germline gene therapy will correct the genetic variants of the reproductive cell of an individual, and this would be passed down to future generations. Benefits of gene therapy: offers cure to several diseases like cancer, Parkinson's disease, AIDS, asthma, diabetes, heart disease, as well as hereditary diseases. The benefits of the gene therapy is to give someone that is born with a genetic disease or who develops cancer the chance at the normal life. Disadvantage of gene therapy: modifying organism means modifying its capabilities. Viral vectors could recover its ability to cause disease. Also, it can damage the gene pool, result to multi-gene disorder. High cost and ethical issue. So the disadvantage of gene therapy is to find a gene that is not functioning right and to insert the healthy portion into the gene.
Biotechnology. So what is biotechnology? Biotechnology is a technology that use of biological systems, living organisms, or derivatives thereof to make or modify products or processes for specific use, like brewing and baking bread are one of the examples of processes that fall within the concept of biotechnology by the uses of yeast. So, yeast is the commonly na siya madungog mo na siyang ginasago sa bread and pancake para magtubo or expand. So, yeast is very useful for living organisms. It is used to produce the desired product. So, when you say to produce the desired product, it describes any goods or service that give high satisfaction and long-term benefits. Genetic engineering. So, genetic engineering is a technique that allows genes in DNA to be transferred from one source to another, leading to the production of living modified organisms or genetically modified organisms. Genetic engineering, also called genetic modification or genetic manipulation, it is indirect to manipulate of an organism's genes using biotechnology. So, it is a technology that used to alter the genetic makeup of an organism. So, when you say alter, it is making different without changing into something else. Next is genetically modified organisms. A genetically modified organism is an animal, plant, or microbe whose DNA has been altered using genetic engineering techniques. So, here are the examples of GMO nga na-altered na gamit ang genetic engineering techniques. First is the Bacillus thuringiensis corn or the BT corn. Second one is the genetically modified papaya. Third is the Roundup Ready soybean. Fourth, golden rice. Fifth, Arctic apple. And last is the Aqua Advantage salmon. So, ang mafamiliar lang kay nato diri no kay kaning BT corn o golden rice. So, BT corn, or also known Bacillus thuringiensis, is enhanced through biotechnology to protect against insect pests. It is built-in insect protection comes from naturally occurring microorganisms called Bacillus thuringiensis or BT. And next is the golden rice. So, golden rice is genetically modified to produce beta-carotene, which is not normally present in rice. Beta-carotene is converted into a vitamin A. When metabolized by the human body, we need a vitamin A for a healthier skin, immune system, and vision. So, what is beta-carotene in golden rice? So, beta-carotene is a compound that gives vivid yellow, orange, and red coloring into crops. So, otherwise, biotechnology uses beta-carotene to produce golden rice. Benefits of GMO. So, here are the following benefits of GMO. And some benefits of genetic engineering in agriculture are increased crop yields, reduced cost for food production, reduced need for pesticides, enhanced nutrient composition and food quality, resistance to pest and disease, greater food security, and medical benefits to the world's growing population.
Potential risk of GMOs. Number 1. in a decade of study in the effects of GMOs to human and the environment. In studying GMOs, we must assure the quality of GMO's finished product. Failure of transmitting gene or DNA may cause harmful effects to human and also in our environment. Number 2. Promote mutation or in organism, which the long-term effects are still unknown. In the last 2 years until now, we face the pandemic. This pandemic were called coronavirus disease. These viruses are example of organism that mutate into another organism or a high-class virus. This kind of organism continuously mutating, and the effects of the said organism are continuously upgraded too, where we are also don't know how long it costs to human being.
Number 3. Human consumption might have the following effects: more allergic reaction, gene mutation, antibiotic resistance, change in nutrition value.